Today we are going to start the sixth chapter of biology, that is enzymes. In this chapter we will study that what actually the enzymes are, how they will work, what are its characteristics, what are the factors that are affecting enzyme action, what is the mechanism of enzyme action, its specificity, what are the uses of the enzymes. So the first thing is about the introduction of the enzymes. Enzymes are the biological catalyst, and their main function is to speed up the rate of the biochemical reactions. Most enzymes are the three-dimensional globular proteins, so their structure is tertiary and quaternary structures. Some special RNA species also act as the enzymes, and these are called as ribozymes. For example, the hemorrhoid ribozyme. They also act as they are actually the RNA species, but they act as the RNA enzymes. So the active site of an enzyme is the region that binds substrates, cofactors and prosthetic groups and contains residue that helps to hold the substrate. That means the active site is that part on which the cofactors or substrates are getting attached at that point. Then the active sites generally occupy less than 5% of the total surface area of the enzyme. Active site has a specific shape due to the tertiary structure of the protein. The change in the shape of protein affects the shape of the active site. If the protein, the shape of the protein affects, that means the active site and the function of the enzyme will also be affected by it. What is the active site? Active sites can be divided into further two types, that is the binding site and catalytic site. Binding site is what? It chooses the substrate and binds it to the active site. Means the binding point where it gets binded, that is the binding site. And then catalytic site, it performs the catalytic action of the enzyme. The process, all the catalytic actions of the enzyme will be to the catalytic site. Here is the figure as well. So you can see that it is the inactive protein, this is the active protein, this is the active part of the protein. Here the binding proteins are attached, and there is the binding site over here where the enzymes get attached. So cofactor is the non-protein molecule. It is not the protein one, it is the non-protein molecule that carries the chemical reaction that cannot be performed by standard 20 amino acids. So cofactors are of two types, that is the organic cofactors and the inorganic cofactors. So if the protein, it is actually the inactive protein, when cofactor gets attached to it, that becomes the active protein, and after its activation the enzyme will bind it here. The inorganic cofactor, these are the inorganic molecules required for the proper activity of the enzymes. The inorganic cofactor, these are the organic molecules that is required for the proper activity of enzymes. There are types of the organic cofactors, which is the prosthetic group and coenzymes. Cofactors can be either inorganic or organic. So if organic cofactors are tightly bonded to the enzymes, they are called as prosthetic groups. If organic cofactors are loosely attached to the enzymes, they are called as the coenzymes. And the coenzymes transport chemical groups from one enzyme to another. So some important vitamins like the riboflavin, thiamine, folic acid, they also act as the coenzyme. So an enzyme with its cofactor removed is called as the apoenzyme, and the complete complex of a protein with all the necessary small organic molecules, metal, ions and other components is termed as the holoenzyme or holoprotein. Then the next term is substrate. Substrate is what? The reactant in biochemical reaction is termed as substrate. Means the reactant, if the reaction is taking place, the reactant is the substrate while the product is the enzyme. When the substrate binds to an enzyme, the reactant is biochemical reaction that takes place is the substrate, while when substrate binds to the enzyme, it forms the enzyme substrate complex, which is actually the product of enzyme and substrate. So sites of enzyme synthesis, that enzymes are synthesized by ribosomes, which are attached to the rough endoplasmic reticulum. Information for the synthesis of enzymes is carried by DNA. So amino acids are bonded together to form the specific enzyme according to the DNA code. Here is the endoplasmic reticulum and the ribosomes are here. Ribosomes do what? These get attached to the rough endoplasmic reticulum and then enzymes are synthesized. Then next thing is about the intracellular and extracellular enzymes. Intracellular enzymes are synthesized and retained in the cell for the use of cell itself. Means it is intracellular, it remains inside the cell, it is for the cells. They are found in the cytoplasm, in the nucleus, mitochondria and chloroplast. Enzymes involved in the reduction in the mitochondria. Then the extracellular enzymes are synthesized in the cell but secreted from the cell to work externally. They are synthesized in the cell but they are excreted out to work externally. So the digestive enzyme produced by the pancreas are not used by the cells in the pancreas but are transported to the duodenum where it starts working there. Now, what are the characteristics of enzymes? The main characteristics of enzymes are that enzymes speed up the reactions by lowering the activation energy of the reaction. Their presence does not affect the nature and properties of the end product. They are highly specific in their action that in each enzyme can catalyze one kind of substrate. The small amount of enzymes can accelerate chemical reaction. That if the small amount of enzyme is present, it can even increase the chemical reactions. Enzymes are sensitive to change in pH, temperature and substrate concentration. That means that if there is a light change in the pH, temperature and substrate concentration, the enzyme activity will be affected. Turnover number is defined as the number of substrate molecules transformed per minute by one enzyme. So catalase turnover number is 6 into 10 raised to the power 6 per minute. Now, what are the uses of enzymes? Their main uses is in the food industry, brewing industry, paper industry and biological detergent. In food industry, the enzymes that break starch into simple sugars are used in the production of white bread, buns, etc. In brewing industry, enzyme breaks starch in proteins and the products are used by yeast for fermentation. Then in paper industry, enzymes break starch to lower its viscosity that aids in making paper, that helps in making the paper. Then biological detergent, that protease enzymes are used for the removal of protein stains from clothes. Amylase enzymes are used in the dishwashing to remove resistant starch residues. This is all about the uses. Then mechanism of enzyme action, that how the enzyme will work. The catalytic efficiency of enzyme is explained by the two perspectives, that is thermodynamic changes and processes at the active site. But there are two different models that were given, according to which we can easily study about the mechanism of enzyme action. The first one is the lock and key model, which was given by Emil Fischer, and the other one is induced fit model that was given by Daniel Koshland. In lock and key model, the lock and key hypothesis assumes the activity site of an enzyme are rigid in its shape. There is no change in the active site before and after a chemical reaction. While in case of the induced fit model, according to the recent studies, according to this, exposure of enzyme to substrate cause a change in enzyme, which causes the active sites to change its shape to allow enzyme and substrate to bind. Means it can change its shape according to the presence of enzymes. That enzyme can easily get binded to it. Here is the structure of induced fit model. Then the next thing is about the factors affecting rate of chemical reaction. That there are three factors: temperature, hydrogen ion concentration, that is pH, and substrate concentration. What is the effect of temperature? Temperature effect, that there is some optimum temperature according to which the enzyme can easily work. Every enzyme works at its maximum rate at a specific temperature, and that temperature is called as the optimum temperature. That when temperature rises to a certain limit, heat adds in the activation energy and also provides the kinetic energy for the reaction. So reactions are accelerated, means the temperature is increasing, the reactions get increased. But till a certain limit, like you can see in this figure, the temperature is rising, along with it the rate of reaction is also increasing. But at a specific height, that is, for example, the 40 degrees centigrade, if it crosses the 40 degrees centigrade of the temperature, then the structure of the enzyme will get ruptured. And why it happened? Because when temperature is raised well above the optimum temperature, the heat energy increases the vibration of the atoms of the enzyme, and the globular structure of enzyme is lost. And this loss of structure is called as the denaturation. It actually results in a rapid decrease in rate of enzyme action, and it may be blocked completely. So the rise in temperature is not good for that. The optimum temperature is good if you have to continue the reaction. The next is about the effect of pH. What will happen in it, that all enzymes work at their maximum rate at a narrow range of pH. In the same way that increase in the pH can cause the increase in the reaction rate, but till a certain limit, that is the optimum pH. The most enzymes exhibit optimal activity at pH value between 5 and 9. So higher or low pH value, then optimum value will cause ionization of enzyme, which results in again the denaturation of enzyme. Here is the structure figure given about the different type of enzymes like pepsin, what happened in the case of pepsin, trypsin, arginase, its activity that is affected by the pH. Next is about the substrate concentration. Substrate concentration means if enzyme molecules are available in the reaction, it increases in substrate concentration. Obviously, its increase causes the increase in the substrate concentration. But if concentration of enzyme is kept constant and amount of substrate is increased, then a point reached where any further increase in the substrate does not increase the rate of reaction. Means that if it reaches at that point, the concentration, if we added the substrate in it, there will be no effect on the enzyme, and that we can say that this state is called as the saturation of active site, and reaction rate does not increase, means it stops, it continue with the optimum rate. There will be no effect on the structure, but what happened, that substrate concentration can causes, like you can see in this figure, that the rate increases, but when there is a specific site, we are added the substrate at the specific point, it reaches that either we add substrate or not, there will be no effect on the enzyme reaction. So that is called as the saturation point. Then there is next thing about the enzyme specificity. Specific enzyme is for the specific substrate, or we can say specific substrate is for the specific enzyme. So enzymes are highly specific in nature, interacting with one or few substrates and catalyzing only one type of chemical reaction. Like it interact with different substrate but catalyze only one type of the chemical reaction. So substrate specificity is due to complete fitting of active site in substrate. There is example that oxidoreductase do not catalyze hydrolyzed reaction, and hydrolase do not catalyze reaction involving oxidation reaction. Like specific enzyme have a specific function. This is all about the enzymes.